Welcome back to the Digestive System podcast. In this section, we are going to cover the vertebrate gastrointestinal system and how it functions. Okay, so the structure of the gut is very important to know. The lumen is the cavity of the gut. Mary, what's the mucosal epithelium?
It's the first layer of cells in the lumen, and it has microvilli and absorptive functions.
Why are the microvilli so important again?
Because surface area.
Good. Greta, what's the submucosa?
The submucosa contains blood and lymph vessels and nerves have sensory functions.
Really, nerves have sensory functions?
Yeah.
Okay, so there's a lot of mechanical activity involved in moving food down to the stomach. Swallowing is actually a complex series of reflexes initiated when the tongue pushes the food to the back of the throat. The esophagus carries the food and water to the digestive system. Another very important piece of this process we've already talked about is the epiglottis, which prevents food from going down the trachea. Food and water from going down
So that's
the
like
trachea.
where we get the saying, don't let it go down the wrong pipe. So,
Yes. This diagram is very self-explanatory. Yeah. So, peristalsis is the action of the muscles contracting and relaxing to move the food down. So one part will contract, the next part will relax to allow the bolus of food to go down to that section. Then that contracts, so on and so forth. But relaxation, where the food moves to, is called the anticipatory wave of relaxation. This is what prevents the food from moving back up that esophagus. Mary, what are sphincters? Okay, you don't know. The circular muscle openings between different parts of the digestive system. Two important ones to know are the lower esophageal sphincter, which is at the junction of the esophagus and the stomach, and the pyloric sphincter, which is between the stomach and the intestine. Chemical digestion begins in the mouth with the enzyme amylase.
MCAS throwback, because that open response.
Yeah. Good times, guys.
It hydrolyzes bonds between glucose monomers that compose the starch molecules. The main role of the stomach is to store food and make digestion slower than ingestion. However, the stomach does have HCl and some other enzymes that begin the digestion of the food. There also has gastric pits, which are literally like infoldings or pits that are inside the stomach that have three types of secretory cells that secrete different enzymes. The chief cells, the parietal cells, the mucus secreting cell, and the mucus secreting cells. The chief cells secrete pepsin, which is a proteolytic enzyme. It is secreted as the zymogen pepsinogen. Here you can see how pepsinogen is activated to become pepsin, and HCl is also released from another type of cell, the chief cell. Autocatalysis is the positive feedback process which causes the activation of pepsin, because the activation of pepsin causes the activation of more pepsinogen, so more pepsin is created. Here's a closer look at the parietal cells. You can see the different exchanges. I am going to leave this slide for a couple seconds so you can pause the video if you need to help understanding it better. It shows the potassium going in and out of the stomach and how the Cl ions and H ions enter the stomach and then they're combined in the stomach to form HCl. You can also see the H2O and the CO2, which is obtained from the blood vessel. And HCO3 is how the body transmits CO2 back into the environment, into the lungs, where it's excreted. Chyme is the mixture of gastric juice and partially digested food in the stomach. It's pushed to the bottom of the stomach to the pyloric sphincter with a series of contractions. At each contraction, the sphincter relaxes just a little to allow a little chyme through at a time to prevent the small intestine from being overwhelmed right after a meal. The stomach also acts as a buffer. Here you can see the gastric pits, literally holes in the side of the stomach that are lined with all these secretory cells. The small intestine is called the small intestine because of its diameter, but it's actually almost 6 meters long. It continues the digestion of carbs and proteins and begins the digestion of fat. It also begins the absorption of nutrients. Mary, the folds, the villi, and the microvilli provide a large what?
Surface area.
It's bold and underlined because it is
It's
very, very, very important. The duodenum is the first part of the small intestine, and the majority of digestion occurs here. The jejunum and the ileum is where most of the absorption occurs. Mostly 90% of absorption occurs in these two parts of the small intestine. The liver, disgusting food, but it creates bile from cholesterol. This bile flows through the hepatic duct and the gallbladder stores it. When fat enters the duodenum, a hormonal signal is released, which causes bile to be pushed into the small intestine, where it separates fat molecules into smaller fat molecules called micelles. Here's an example. Here you can see the bile being created in the liver and flowing through the hepatic duct, and that little small part with the arrow on both sides.
So Joel, what's that blue thing?
What blue? That blue thing is the pancreas actually.
Oh, the pancreas.
Yes.
Which we will cover
The
soon.
common bile duct is what's shared by the gallbladder and the liver, and it flows into the small intestine. The pancreas are both actually an exocrine and an endocrine gland. The exocrine part produces lipases, amylases, proteases, and nucleases. Some are also released as zymogen. Another enzyme releases trypsinogen. Active trypsin can cause the release of more trypsinogen, which will in turn be activated even by enterokinase. It's very dangerous because if the trypsin are activated early in the pancreatic duct, this can lead to the annihilation of the pancreas, which a lot of organ death. Here's a comprehensive digestive system image so that you can look through the whole thing and try to understand more what I've been saying in words. Absorption is where peptidases divide polypeptides into absorbable sizes. Solvent drag is water traveling between cells that carries the nutrients, so in the extracellular matrix, not through the cells. Chylomicrons are triglycerides that are combined with cholesterol and phospholipids with a protein coat that makes it water soluble. The absorbed nutrients go to the liver, and water and ions are absorbed in the large intestine. The colon is another name for the large intestine. Water and ions are absorbed. It also creates feces. Constipation is a result of too much water, and diarrhea is a result of not enough water absorption. That's mixed up.
No, that's right.
Wait.
That's right. Joel doesn't know what he's talking about, guys.
Yeah, not really. Just kidding. What the cud? So, ruminants are cud chewers. They have a four-chambered organ. They have four-chambered organ to digest their food because they're mainly herbivores. The first two chambers are called the rumen and the reticulum. Here, microorganisms break the cellulose down with fermentation. Then the cud is regurgitated, then chewed again and re-swallowed. The nutrients are metabolized and absorbed during the second swallowing. Here's some cuddy vocab. The omasum is where water absorption occurs, which concentrates the partially digested food. The abomasum is where microorganisms are killed by HCl, the bad ones, the mad microorganisms. Some other vocab. This doesn't really relate to ruminants. The cecum is microbial fermentation chamber that empties into the large intestine. An appendix is a vestigial version of a cecum in humans. Coprophagy is where cecil feces are re-ingested from the anus to absorb the leftover nutrients.
Wow! I love knowledge.
Next section. How is the flow of nutrients controlled and regulated? There are many hormones, one of which, including secretin, stimulates pancreas to create a solution that has many bicarbonate ions. Cholecystokinin stimulates the gallbladder to release the bile and pancreas to release digestive enzymes. Example, this is when fat enters the small intestine. Gastrin is released from the stomach into the blood, and it creates when cells in the lower stomach recognize the presence of food, the feeling of satiation. After you eat, there's two periods: the absorptive period, where nutrients are being absorbed, and the post-absorptive period, where energy, metabolism, and biosynthesis just run on your internal reserves. So post-absorptive doesn't always happen after you eat. You may eat before it happens. The liver acts as a traffic director. It has many functions. It converts the monosaccharides into glycogens and fats, or vice versa. Gluconeogenesis is the conversion of certain amino acids and other molecules into glucose.
Dana Mackey, if you are in the building, or any of the buildings, please come to the upper school front desk. Dana Mackey, upper school front desk.
So apparently Dana Mackey needs to go to the front desk. So, lipoproteins solve the problem of transporting fats. They have a hydrophobic fat core, and their cholesterol with a hydrophilic protein cover that allows them to travel in water. Lipoproteins solve the problem of transporting fats. They have a hydrophobic fat core and a hydrophilic protein cover that allows them for suspension in water. Chylomicrons are the largest lipoproteins and are produced by mucosal membranes. Insulin is released by the pancreas when blood glucose rises during the absorptive period. It helps with the uptake, utilization, or storage of glucose. It acts in skeletal muscle, not muscle, adipose tissue, and the liver. Diabetes is when the pancreas can't produce insulin. Diabetics have to get their test their blood a couple times a day to check their sugar and inject insulin depending on when they have meals. There are different densities of lipoproteins which affects their functions. High-density lipoproteins, or HDLs, remove cholesterol from the tissues and transport it to the liver. Low-density LDLs, or LDLs, transport cholesterol where needed for biosynthesis and storage. And very low-density lipoproteins is VLDLs, and they transport mainly triglyceride fats to fat cells around the body in adipose tissue. Here is a flowchart that shows what happens in the body when there's glucose or when there isn't glucose. So if there's a decrease in glucose, the pancreas secretes glycagon, which is increasing the circulating glycagon, breaking down the glycogen in the liver, and a release of glucose to the blood. There's an increase in blood glucose, pancreas create insulin, the insulin is now circulating. There's an uptake of glucose in the cells for metabolic energy, fat synthesis, or glycogen synthesis, and it decreases the blood glucose. Intake is regulated by sensations of hunger and being satiated. Satiated. It is influenced by the hypothalamus. I'm going to ask you to look in your books to define leptin and ghrelin. We're now in the final stretch with the last section about ingested toxins. Thank you for your enthusiasm, Grandma.
Yes.
Okay, so we're just going to do a quick overview of this. The field of studying poisons is called environmental toxicology. Most synthetic toxins cannot be metabolized. Bioaccumulation is when there's a high concentration of poisons in predators that eat contaminated prey. That's all for That's all for the digestive system, and make sure you fill out that form. Will be worth somewhere between 10 and 20 points on your quiz.